Hi, everyone. I'm Megan, science advisor, co-chair of the executive committee of the DNA sequencing group. And what I thought I'd talk about tonight is what we've kind of found from what we did in Sequanota 2023. Well, I say we, that's a broad we. It was mostly done by John Plischke here on the left and Stephen Russell on the right. Stephen came as one of the guest mycologists, and I think Saranel is going to talk about a bit of the work that he does as well, but he's really been this prolific force of nature in sequencing macrofungi in North America. So we were really lucky to have him come to Sequanota. He and John spent hours and hours and hours during the foray collecting samples, drying them down, and then he took them back to his lab and did nanopore sequencing and posted the results to iNaturalist. So somewhere around August of this year, in 2024, we got the results, and I've been kind of going through them. As you might imagine, they took about 600 samples, and there's a lot of results to go through. So I'm just going to provide sort of an overview, a higher level overview. Every single name has a story to tell, and maybe we can come back and tell some of the more individual stories later on in the year and going into next year when we'll have some 2024 results. So what usually happens on a foray? Usually on a foray, you have happy people that go out into the woods and they collect mushrooms and they bring them back and put them somewhere on a table in baskets, and people who know what they're talking about come along and put names on the fungi that they're seeing. And everyone oohs and ahs, and you have a table talk. And at the end, you come up with a list of what you saw. And that is usually how it happens at most sort of mushroom club forays. There are records in literature for forays that happened with the Mycological Society of America, for example, where they would go out and collect everything, attribute the IDs to whoever was there and made the ID, and actually save things for voucher specimens. So if you look at the 1952 foray from the Mycological Society of America, they took all of those samples, they were in Ithaca, and those are stored at the Cornell Herbarium. But most mushroom clubs, you don't really store your samples. Everyone is starting to document things on iNaturalist, so we at least have the documentation. So these are some of the pictures from Sequanota 2023. Lots of really good stuff, really nice images. And so at least documenting things on iNaturalist has been an improvement to just having a list of names that aren't tied to anything, no voucher, no sequences, nothing to actually corroborate that that's what you saw. So in 2023, there were 2,056 observations in iNat, which is excellent. That really gives us a lot of good images that go back to what people were seeing. Of the various fungi that were brought in and put on tables, like I said, John Plischke and Stephen Russell took about 600 vouchers for sequencing, 640. And of those, about 500 successfully produced sequences, and these are ITS sequences. Of those 500, 54 agreed with confirmed field ID. So someone went out with iNaturalist, gave something a field ID, it got taken in for sequencing, and the sequencing fully agreed with that field ID. 407 of them refined the field ID, and that might be overstating it a little bit because I think because things were being taken for sequencing, people might not have spent as much time making that field ID as they would have otherwise. So even IDs that were made by John Plischke or Martin Livesey or other people who really know what they're doing, they just put in inosity or amanita. And I think part of it was because they knew that sequences were going to be generated. So that 407 is probably an overstatement. Had those folks actually made a field ID, we probably would have had more agreement. And of those, 36 were novel in some way. And in a lot of cases, you'll see provisional names in the next few slides, which are sort of new. And when I say novel in some way, it was the first sequence ever anywhere in any DNA repository, or one of the first three, or something that just seemed really, really brand new and different. And 136 were attempted but failed, which is how it goes with DNA sequencing. So, like I said, over the years, we've put together a MAW life list, or the people who, mostly Mitch, has put together a MAW life list and has done a lot of the work to make sure that this is annotated and stored, and that we have a list of what people have been finding at Sequanota. So thank you, Mitch, for all that work. And that at this time has about 709 names. So of the 709 names on the list, we supported 71 of those names with vouchered sequence specimens. So we know that those names are associated with specimens that we can go back to, sequences we can go back to and compare to things that we find in the future. We also, through the work that John and Stephen did, 270 names were added to the MAW life list. So if you look at the life list now, plus everything that's in iNaturalist from Sequanota 2023, that's over 900 names associated with Sequanota. A lot of them are provisional sequence-based names that I'll show you in a minute, but that's pretty impressive that we're up to about almost 1,000 names associated with Sequanota. So Tom said a lot of names, Mitch said a lot of names, and a lot of people nowadays are really nervous about saying anything more than maybe Amanita or anything more than Russia, because DNA sequencing has changed so many things and is changing the names and the taxonomy of a lot of fungi. But don't worry, most of the things that were ID'd that are common edibles that everyone knows, or things that are toxic, like scleroderma, these IDs were confirmed. So Stropharia rugosa annulata was found, confirmed, supported. Sparaxis spathulata, also confirmed. And there were a number of other species like that. So there were a lot of field IDs that you can make that are fully confirmed by DNA, and you don't have to worry about those. But I was going to talk about some of the other things that were found. So Mitch showed one of those pictures of Amanita and the Amanita vaginata group. He mentioned the fibrils on the edge. A lot of them are sort of grayish. So these are 4 species in Amanita, and sequencing gave us provisional names for these 4. So on the original list from Sequanota, we have about 28 names of 28 names of Amanita, which expands to 50 names if we use the sequencing results. And just to show some of these, so this is Amanita finis provinciae. This is the 3rd known sequence of this Amanita in North America. There's the INAT number if you want to go and look at it. And these names in quotes, a lot of them come from people like Rod Tullis and others who are working in these different genera. So sometimes you'll see a name that associates with a location. Sometimes you'll see a name like this. This isn't a published name yet, but it's a name that's been consistently associated with a certain morphology. And in this case, this is the 3rd known sequence of something with that morphology in North America. Another one is this Amanita spa PA03, PA for Pennsylvania, because it came from Sequanota. And the sequence of this was new to any DNA repository in the world. So it's an unassuming little mushroom. You look at it and say, yeah, that's an Amanita in the Amanita vaginata group, and that's about as far as we can go. But having this DNA sequence means that if we see something similar next year, we can collect it and sequence it and see if we found the same thing. And I think that's really part of the power of having the voucher specimens and the sequences. Another one is Amanita homolytineni, which is widespread in the eastern US. There are dozens of sequences from throughout the eastern US, and that is something that we also have at Sequanota. And here's another specific Pennsylvania novel one, Amanita spa PA04. And as of today, there's only one sequence in any DNA repository of this one as well. So instead of guessing, do we have the same sort of Amanita vaginata, Amanita in the vaginata group? We can now hang our hat on 4 separate sequences and going forward, see whether we repeat finding those again. Clavulinopsis, we had 3 names on the original list, and that is now 10 names if we include the sequencing results. And that's things like Clavulinopsis aurantiocinabarina, Indiana 2. There are only 2 sequences of this one in all the databases. Clavulinopsis aurantiocinabarina, Pennsylvania 1. This is another one where the only sequence in all DNA repositories now comes from Sequanota. So it's a newly sequenced species. These are Clavulinopsis lenticolor, Indiana 3. And it was Indiana 3 because it was the 3rd sequence in Indiana that was associated with the Clavulinopsis. But there's only 1 Indiana sequence, and there are 2 sequences from Sequanota. This one is tiny. You can see in the above picture, there's the voucher label, and there's the Clavulinopsis, which is just millimeters high, hanging out amongst the leaf litter. So tiny, but mighty in the fact that it's, again, something new. Portnarius is another one where we expanded the list of names quite a bit. There were 12 names on the original list and 30 names in Portnarius found from Sequanota if we include the sequence results. That includes this one, Portnarius Indiana 08, which is widespread in Eastern US and Canada. There were 10 collections made at Sequanota. And having all of the INAT results plus the DNA results really lets us see some of the variation in morphology. I'm not sure I would look at all of these and call them the same thing. They do have quite a bit of variation. You have one that's got a really bulbous base. Of course, when they dry out, being hygrophorous, they look a little bit different. But that really, having the sequencing behind it really gives us some confidence and a lot more information about what's going on. We also had some novel Cortinarius Indiana 128. And I don't know how many he's up to for Indiana now in terms of Cortinarius, but there is 1 sequence of this from Indiana and 1 sequence from Pennsylvania, from Sequanota. We also confirmed a few things. So for example, what we've been calling Cortinarius iodes was confirmed to be Cortinarius iodes. So that's also reassuring. And just to kind of, again, overview, I mean, each of these names, you could really spend a lot of time talking about the new species, how the genus was described to begin with, what goes into having that description, what the new species represent. So in Entoloma, we doubled the number. There were 11 on the original list. That becomes 25 with sequence results. Geoglossum, there were none on the original list, and I'm not sure why. I wondered whether that was a mistake, but it becomes 4 with sequence results. We added 6 hygroscopy names to the original 13, 12 inoscopy names to the original 8. We had quite a few Lactarius or Lactifluus on the original list and added 12 to the 35 and confirmed 5 of the original names. And for Scutellaria, there were 3 on the original list and 7 names were added. So those were some of the ones where we really added quite a lot. And then there are a lot of singletons kind of throughout of new things. And then we come to Russula, and I forget what Tom called it, just another red Russula, I guess. We've decided, perhaps I decided as a club to focus on Russula as one of the many things that we're focusing on, partly because they are really, really hard to identify morphologically. So we had 28 names from Sequanota on the original list, and of those 28, we confirmed 2 of them and added 38 names. 65 specimens were sequenced to add to those names. On the bottom there, I have a quote from Michael Kuo, and if you haven't gone to look at his page on Russulas, it's really entertaining and very to the point and on spot about how hard Russulas are to identify. And so he says, identifying Russulas is a lot of work, and I wouldn't blame you at all if you decided to simply call every red Russula you find Russula emetica, since that species is found in most field guides. And who knows, maybe we will find out that they really are all Russula emetica once their DNA is studied. though it is equally likely we will find out that Russula medica is the same as some jelly fungus, while there are actually 23 very genetically distinct but morphologically indistinguishable species currently going under the name Russula virescens. Well, not all red russulas are Russula medica, and there are a lot of Russula virescens these days. So he was right whenever he wrote that. So here's a slightly, oops, go back one. A somewhat more interactive part. How many species do people think are present on this slide? How many Russula species? You can put it in the chat and we'll look at it later. It could be as many as 12. Someone said 5. No one else wants to venture a guess. So this is actually 7. There are 7 species here. The ones that are separated at the top are 4 different species. These 2 are the same species. These 2 are the same species. And these 4 represent the same species. But again, this just illustrates how ridiculous russulas are and how they've all decided to hide in the forest in the same way, which is not very well because they have bright red caps and white stipes. But for whatever reason, convergent evolution has meant that all of these russulas have decided to have the same coloration. I guess it works for them, but genetically there is still separation between the species. So of the 65 samples that were sequenced, one thing that you can do once you have those DNA sequences is you can line all of them up in various types of software. So this is an alignment of all of the Russula sequences from Sequanota and some from Maryland that we've also been sequencing as part of the club. And this also illustrates why the ITS region has been used as the barcode region. So if you look in the center is all gray, and that means that these are all very similar. You see colors where there are differences in nucleotides. So you might have an A in one sequence and a T in another sequence. But in the center, they're all pretty much the same. And that is part of that DNA sequence is protein encoding. So that encodes a protein that is part of the ribosome. On either side of this protein encoding sequence is the ITS1 region and the ITS2 region. And those are regions that don't encode for proteins, but they still get transcribed. And that's why it's so useful, because you have this anchor point in the middle, and then you have these hypervariable regions that can differ between species. So you can make an alignment like this and have a look at how the nucleotides differ between the species, and then also use software programs to create phylogenetic trees that also help you show how these different species cluster together. So in this case, starting from the top, you've got Russula pulchra, and those are 3 different iNaturalist observations with that name on them, and they all cluster together as they should. You've got the various provisional names that have been put on some of the other iNaturalist observations. And I was just going to show you those 2 that have arrows in the center. So once you've got them in a phylogenetic tree like this and clustered together, you can go back and look at the individual samples that were taken. And in this case, it's hard to say. These are all Russula spa Indiana 114. If I came across those 4 in a forest, I don't think that I could say that they're the same species. But it allows us to at least start looking, are there small differences? Is there anything that we can use? Or maybe these all have some sort of funny shaped cystidia. Or maybe they all just look the same and we have to do sequencing forever and ever in order to get to species ID. Same with Russula spa Indiana 129. One of the things I really found interesting about this is the one at the very top that is all whitish to yellow. So, we have albinism in other organisms, and changes in color have been documented in fungi. So maybe that's just an albino member of this species. The cap color does change quite a bit. You've got some that are very, very cherry red and some that are more of a peach pink. And maybe if we sequenced more gene regions, we would see that these are actually even more, that we could split this even further. But for now, these are all in Rushlispa, Indiana 129. So what? I provided just an overview of some of the things that we've found so far. There's a lot more to do. I showed that alignment for the Rushula. An alignment like that can be generated from pretty much all of the genera that were sequenced, adding in sequences from other places, from GenBank, from MicroMap, from other studies. And that gives us a scaffold that we can start to put our new sequences on, and we can really get a better idea of just how many different species we're finding out at Sequanota. Another thing that this allows us to do is to go back. So we know where each of these were found. These are all associated with iNaturalist records. We can go back to pinpoint the exact location that this one was found in and see if we find it again. And so for all of the more interesting, novel things that came out of the sequencing, we can go back to that location and see, do we see it again next year? If not next year, the year after. And having the sequences and the iNaturalist observations will really let us know whether we're finding the same thing over and over again. In 2018, Richard Jacob found this fungus. They called it at the time hygroscebe, Calyptriformis. And he found it in one location in Sequanota, took a picture, put it in iNaturalist. And then in 2023, went back to that same location and found it again. In that case, it happened to be Sequanota 2023. So a specimen was taken and sequenced. And it is being given a provisional name of Porpulomopsis atlantica. That's not been published yet, but these apparently are different enough from the Calyptriformis that they're being given a provisional name. So that's sort of the power of having the iNaturalist observation and the sequence. And I look forward to having more stories like that in the future from some of the other genera that we've been sequencing. So I propose that we make Sequanota a big scavenger hunt. We have a list of 600 and some names that we have not supported with DNA sequencing. So we know where some of those are. We can go out and find them again and really support those names of DNA sequencing. And we can go back and see if we can find some of the new things again. And I can't wait. Looking forward to it next September.
I feel like you, Megan, just gave us a look at what we've been doing. And what I would love to do is give us a look into what's the future of the DNA research committee. So we have embarked into an initiative in collaboration with the Mycota Lab and Stephen Russell, as well as Parks and Recreation, to investigate what is living in the soil. So this is being done through environmental DNA. And what is the DNA, you may ask? What is the eDNA, you may ask? Well, it's just the genetic material that any organism sheds into the environment just by living in there. You don't necessarily have to see the actual organism to be able to detect it. We all live our cells. It could also be fragments of an organism, anything that's produced that still have DNA in it by any creature can be detected by eDNA. And it's eDNA in the general terms can be collected from water, soil, or even air filters. Um, from the point of view of why Parks and Recreation would be interested in this is of that conservation. We really want to see what is living in the parks and the land of Parks and Recreation, and um, there could be measures and policies that could be affected by the detection of threatened or sensitive species. So we really care about what's living in the soil. So the site that was selected for this particular initiative was Patuxent River Park. This is a really extensive piece of land that also gives you a variety of habitats or ecosystems. Like I said, through the Natural and Historical Resources Division, the division I work for, we were able to obtain permissions to go and collect in park property. And this property is in the skirts of the Patuxent River. Part of this property also gives us the opportunity to see two very distinct ecosystems. You have the upland forest, which was used for sample number one, and you have the tidal marsh area, which was used for sample number two. When you look at these two sites, let me see. Yeah. Okay, I'll talk about them. In any case, um, it's a pretty important park when we look at the ecology of the region. Not only do you have different ecosystems, but it is a critical area for migrating birds in the region. It's a wetland that is the spot for migrating birds, kind of the gas station where they can stop and refuel in their way to both south and coming up north during the spring. So once we registered and agreed to join this adventure with the Mycota Lab, we were provided a kit where we received the equipment for both in the field and in our lab. So basically, we got flags to set up the samples and tools to collect the soil to give us the same amount of soil for every subsample, as well as the instructions to upload this into iNaturalist, which is the data depository for the eDNA project at large. So once we get the kit and we decided where we were gonna take those samples, one was upland forest. And that was a pretty interesting, a pretty interesting excursion into that particular part of the park. It's beautiful. It mixed hardwoods and what struck me the most was the presence of lycopodiums or it's a type club moss. The club mosses, it's a type of plant that requires a lot of time to grow. It grows very slowly, and it's usually a good indicator that there's high diversity and the soil has been quite stable for a while. Um, and I had never seen a lycopodium forest. It was just so in such high quantities that we definitely needed to stop right there. And when you looked around that particular site, you also saw some beech drops, which is a plant that is parasitic with beech trees, and crane fly orchids. There's a typo there. Anyway, um, all three species of plants are indicators of high diversity in the soil because they require a stable soil and the hosts of all of the species involved in that symbiosis to be there. So really good indicator of high diversity. In the case of sample number 2, we selected the tidal marsh. In that area, we also had the presence of old swamp cypress trees and other native plants that is also a good indicator of high diversity in that particular ecosystem. And then when we were in the march, we selected that particular spot because we had more soil than water. It facilitated the collection of that particular sample. But that sample was close to one of the biggest trees in the area, which will allow or support a higher diversity in that particular soil. And if you look at the illustrations at the bottom of the pictures, you can see how the sample is supposed to be set up with one or flag number 1 in the center, and then you go to the 4 cardinal points away from that center at the same distance. In the case of the forest, we really needed to use a mullet to collect the sample because the soil was harder to get that sample um, extracted from. But in the tidal marsh, it was really nice. So no additional force needed. In any case, collect the sample, put it in a bag, put it on ice, and bring it to Mount Rainier Nature Center. And in here, both myself and Marilia Solis had the pleasure to process those samples, which involved sifting them to extract rocks and bigger particle pieces. And then those sieved soils were dehydrated at 65 degrees Celsius and then processed some more and put into tubes that were 35 milliliters, and then we sent them away. So right now we're just praying that they get there safe. And we can't wait to see the samples or see the sample data. When we look at the the actual mushrooms in a particular area, we are so bound to luck and seasonality that the diversity that we can see, it's really minimal. Doing eDNA research allows us a much better look at what's really living there. Um, not We don't have to wait for rain, or we don't have to wait for that specific fruiting period, specific fruiting period, we can just take the DNA out and see what's there. And I can't wait to see all the mysteries that the soil holds and understand better the ecology of that park and hopefully the ecology of our region. Thank you very much for your time. Don't forget to join us this Sunday at Mount Rainier Nature Center for our DNA Research Day. If you are bringing samples to process, please arrive close to 10 a.m. or at 10 a.m. And if you just want to chat and see what's going on and learn more about the kingdom fungi, please come at 1 p.m. while the PCR machine is running. Thank you so much.
Wonderful. Thank you, Megan. Thank you, Sarah and Nella. That was good stuff. And I'm eager to hear more about this eDNA and what secrets we find in the soil. Very good presentation. Okay. I don't see any questions in the chat. We're just about right on time. So unless I see something pop up, I think we're going to call it a night within the next minute or two. And we'll be back in a month from now talking lichens, my favorite. All right. Thanks again for everybody. Have a good evening. Thanks, Sarah, Nella, Megan, Mitch, everybody.